this video we will discuss about Cadherin-1 and Cadherin-3 impact cell invasion in the pancreatic cancer cells. So before we directly go into the topic, we will understand the expression of Cadherin-1 and Cadherin-3 in pancreatic cancer cell lines and also in the patients. So first they validate this, they check the Cadherin-1 and Cadherin-3 in these BXPC3 cell lines, and they are able to see both this Cadherin-1 and Cadherin-3 presence in this BXPC3 cell lines. So once they have the cell lines or model which expresses both Cadherin-1 and Cadherin-3, what it is, they knock down Cadherin-1 and Cadherin-3 and prepare a stable cell lines which is devoid of either Cadherin-1 or Cadherin-3 in BXPC3 cell lines, so that they can study the impact of absence of Cadherin-1 and Cadherin-3 on invasions. And apart from generating these stable cell lines, they also check the Cadherin expressions in the patient samples in the primary tumor tissues using CRCM110 and CRCM08. These are the two names that they have given to the patients. So in this CRCM110, they are able to see both Cadherin-1 and Cadherin-3 presence, whereas in CRCM08 they are able to see only Cadherin-3 presence. So they will use these primary tissues to validate their in vitro findings that they get from BXPC3 cell lines. So first they check their spheroids formations, or how compact these cell lines will be able to form spheroid. That will shows the impact of Cadherin-1 and Cadherin-3 on cell-cell adhesions. So when they check this BXPC3 cell lines that has both Cadherin-1 and Cadherin-3, they are able to get a impact the spheroid formations. But in the absence of Cadherin-1, they do not get a compact spheroid formations. And in the conditions where Cadherin-3 is knocked down, they in this case also they get a complete spheroid formations. And when they check the spheroid formations or spheroid area in the CRCM110 or CRCM08 primary tissues, they are able to see that the CRCM110 has the good spheroid area formations. It is similar to the BXPC3 cell lines, whereas CRCM Cadherin-3 positive and Cadherin-1 negative does not have the compact spheroid formations. So this results indicates that it's the Cadherin-1 that plays a major role towards this cell adhesions, whereas Cadherin-3 has less roles towards these cell adhesions. This is what they conclude from these spheroid experiments. And after that, they also check this ratio of Cadherin-1 to Cadherin-3, the spheroid area ratio, and what they are able to see. In this case, they also use this spheroid experiment to check the invasions. This spheroid experiment does not require cell-cell contact for the proliferations. And what they are able to see that at 24 hours, the in vitro actual cells has in vitro BXPC wild type cells has both Cadherin-1 and Cadherin-3 shows more invasions compared to the cell lines where Cadherin-1 is knocked down or Cadherin-3 is knocked down. Since this experiment does not need cell-cell adhesions for the proliferations, they think that it's not because of the Cadherin absence that they shows invasions. It might be because they shows less invasions. It might be because of some other reasons. And when they check the invading assay on the 2D model, in this case also they are able to see that the BXPC3 wild type cell lines has more invasions compared to the case where Cadherin-1 is knocked down or Cadherin-3 is knocked down. And in this case also, since the cell adhesion does not plays in the invasions role, so they point out, they knock down the possibilities that it's the Cadherin-1 or Cadherin-3 knockdown that is causing a less invasions. Whereas when they check the invading assay with this primary tumor cells, that is CRCM10, which has both Cadherin-1 and Cadherin-3, and CRCM-08, which has only Cadherin-3. They are able to see that CRCM-10 gives more invasions compared to the case where CRCM-8, which has only Cadherin-3 positive. So from this and the other results, they conclude that this Cadherin-1 and Cadherin-3 both plays a role towards invasions, and since when the both are present, they are able to see the maximum migrations. And same things they have observed for the CRCM10 also, CRCM10, which has both Cadherin-1 and Cadherin-3. So let me just clarify, just to for the viewers who has a confusion about the results of the panel B and panel C. In the panel B, both of them are present, they see maximum invasions, whereas when only seeing one of them is knocked down, they see a low invasion. So it indicates that since cell-cell adhesions does not play a major role towards these invasions, they they point out that there might be some intercellular changes occurs because of the knockdown of one of the Cadherin, and that might causing this decrease in the invasions. Whereas when both of them are present, they see some synergetic effect that cause maximum invasions. So I hope you understood the impact, how they come to a point that Cadherin-1 and Cadherin-3 impact cell invasions in the pancreatic cancer cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.